today we will start with our new chapter for class +2, that is chapter number 2, and the name of the chapter is the sexual reproduction in flowering plants. So this is the chapter sexual reproduction in flowering plants. So all the concepts regarding the sexual reproduction in flowering plants are to be discussed in very detailed, and step by step we have to discuss all these concepts that are regarding in this chapter. So before starting this chapter, let us know about what do you mean by reproduction. Reproduction होती क्या है? Reproduction is the process of giving birth to the young ones. Reproduction एक ऐसी process है जिसके द्वारा young ones को produce किया जाता है by the process of reproduction. So let us start with the sexual reproduction is the process of development of new organisms from two parents through the fusion of male and female gametes. So sexual reproduction is the process of giving birth to the new organisms from the two parents through the fusion of male and female gametes. So this is the sexual reproduction. Next is the flower is the main structure that concerned with the reproduction. Reproduction flowering plants के केस में जो मेन हमारा रिप्रोडक्टिव ऑर्गन है या मेन स्ट्रक्चर जो है जो कि कंसर्न करता है विद द प्रोसेस ऑफ रिप्रोडक्शन, दैट इज फ्लावर. द रिप्रोडक्टिव ऑर्गन्स और द स्पोरोफिल्स आर प्रोड्यूस्ड विद इन द फ्लावर्स. अब फ्लावर्स के बीच में जो प्रोड्यूस होते हैं दैट इज द रिप्रोडक्टिव ऑर्गन्स एज वेल एज स्पोरोफिल्स, दे आर प्रोड्यूस्ड विद इन द फ्लावर्स. Sporophylls are of two types. अब यहाँ पर जो sporophylls है that are of two types, that is microsporophylls and megasporophylls. Microsporophylls, they are also known as stamens, and megasporophylls, they are also known as the carpels. So these are the main two types of the sporophylls that are concerned with the flower, that is the stamens and the carpels. Carpel is distinguished. Carpel को हम कैसे distinguish करते हैं? Distinguished as a ovary. अब ovary के बीच में क्या है? Ovule, style and stigma. That means carpel that is distinguished in the ovary, and ovary is distinguished further into ovule, style and stigma. Stamen is distinguished. अब हमने पहले किसको distinguish किया? Carpel को. Carpel is distinguished as ovary, and that ovary is bearing ovule, style and stigma. And next is the stamen, and it is distinguished into filament, anther and the connective. So these are the main parts of the reproductive flower. After the sexual reproduction, we will shift to the sexual reproduction in flowering plant. That means we will discuss about the longitudinal section of the flower. That is the reproductive structure of the angiosperm, and flower is the modified shoot. So let us start this reproductive structure of the flower. In this flower, this is the ovary, and in the ovary, number of ovules are present in the ovary, and ovary is directly attached with the stigma with the help of a style. And outside the ovary, there are sepals and number of petals that are attached with the flower, and these are 5 in number, and they are multicolored. Anther is attached with the help of a filament. So this is the longitudinal section of the flower. And after the longitudinal section of the flower, our next main important topic is that is the whole process of the sexual reproduction in the flowering plants can be occurred in the following steps. मतलब जो हमने sexual reproduction जो है वो flowering plants में जो हमने पढ़ी, that means it falls into 3 categories. That is the pre-fertilization, double fertilization and the post-fertilization. These are the 3 important processes that occur in the sexual reproduction of the flowering plants. First is the pre-fertilization, that is the structure and events. The pre-fertilization events can be studied under the following points. अब जो हम pre-fertilization events पढ़ेंगे flowers में, that means that studied under the following categories or under the following points. And the main points are, number 1 is the pollen grain formation, embryo sac formation, pollination, and the 4th one is the pollen pistil interaction. These are main types of the steps that occurred in the pre-fertilization events. how the pollen grain formation takes place in the flower, how the embryo sac formation takes place in the flower, how pollination takes place, and how pollination pistil interaction takes place in the flower. So these are the main steps that occur in the pre-fertilization events. So let us start with the pollen grain formation. That is our main first step, pollen grain formation. That is the male reproductive unit. अब pollen grain formation में सबसे पहले हम जो discuss करेंगे, that is the male reproductive unit. So as all we know that the male reproductive unit that occurred in the flower is the stamen. Stamen is the male reproductive unit that occurred in the flower. A stamen is the male reproductive unit of the angiosperm. It consists of an anther and a filament. That means जो male reproductive unit है flower का, that is the stamen, वो क्या consist करता है? Anther and a filament. And the anther is bilobed. Anther जो होता है, that is bilobed. That means it is divided into two equal halves, and the lobes encloses 4 pollen sacs. और हर एक lobes में जो है वो 4 pollen sacs होते हैं, या उनको हम क्या बोलते हैं? Microsporangia. Pollen sacs as well as microsporangia. That means these are encloses 4 pollen sacs or the microsporangia. Each pollen sac contains number of pollen grains. हर एक pollen sac में जो है number of pollen grains जो होंगे वो प्रेजेंट होंगे. The 4 pollen sacs lies in 4 corners. अब जो 4 हमारे pollen sacs हैं, that lies in the 4 corners of the diatomaceous anther. वो कहाँ अक्कर करेंगे आपके? Diatomaceous anther में. That means 4 pollen sacs lies in the 4 corners of the diatomaceous anther. And what do you mean by diatomaceous anther? उसके लिए आपको ये भी पता होना चाहिए कि diatomaceous anther होता क्या है. That is an anther that contains 2 lobes. ऐसा anther जो क्या contain करता है? 2 lobes को contain करता है, and that are joined together. और वो 2 lobes जो होते हैं, they are joined together by a non-sporogenous tissue. और वो 2 जो आपके lobes होंगे वो एक दूसरे के साथ ज्वाइन तो कर रहे हैं, पर ज्वाइन कर रहे हैं विद द हेल्प ऑफ द non-sporogenous tissue. That is called as the connective tissue, and that sporogenous tissue that is known as the connective tissue. That means that sporogenous tissue connects the 2 lobes. The anther wall is made up of 4 layers of the cells. जो anther lobe है, ये anther wall जो है, that is made up of the 4 layers of the cells. जो anther wall होगी वो 4 cells की लेयर को मिलकर बनी होगी. And anther dioecious by slits to liberate pollen grains. अब जो anther है वो क्या करेगा? Dioecious by slits, और slits होकर वो liberate कर देगा किस चीज को? pollen grains को. That means वहां से जो liberate होंगे वो क्या liberate हो जाएंगे? pollen grains. Next, the main important topic that in the pollen grain formation is the anther development. How the anther took development. The anther development starts from a mass of homogeneous meristematic cells. अब जो anther की जो development है वो कहां से start होती है? Homogeneous meristematic cells. Homogeneous मतलब same type के cells जो है, that is the meristematic cells से शुरू होती है, and surrounded by an epidermis. And that meristematic cells, they are surrounded by epidermis. Epidermis is the outermost covering of the cell. 4 lobes are formed. इसमें 4 lobes बनेंगे, and 4 layers of archisporial cells are differentiated. इसमें जो है 4 lobes बन रहे हैं, and further 4 lobes जो होंगे archisporial cells के, they're further differentiated. Archisporial cells क्या होते हैं? What do you mean by archisporial cells? हमने कहा कि these are differentiated, and archisporial cells जो होंगे they are further differentiated. For this concept, as all we know that what do you mean by archisporial cells? A cell or a group of primitive cells. एक ऐसे cells या group of primitive cells that form cells जो किस चीज को बनाते हैं? further cells को बनाते हैं which spores are developed. जहां से क्या develop होते हैं? spores develop होते हैं. That means archisporial cells जो है वो एक तरह से cells हैं या group of primitive cells हैं जो कि क्या बनाते हैं? cells को बनाते हैं, and उन cells से फिर further क्या develop होते हैं? spores develop होते हैं. That means which spores are developed. Each archisporial cell divides to form 2 types of the cells. हर एक archisporial cell जो है they further divided 2 types of the cells, primary parietal cells and primary sporogenous cells. These 2 types of the cells are formed by the archisporial cells, and that cells are primary parietal cell and primary sporogenous cells. So parietal cells divide several times. अब parietal cell जो थे वो several times divide करते हैं, और several times divide करने के बाद वो क्या बनाते हैं, anther wall को बनाते हैं, जबकि sporogenous cells जो होते हैं undergoes few division. As compared to the parietal cells, sporogenous cells में जो होती है वो fewer division होती है and forms the microspores, मतलब वो कम spores को develop करते हैं, और pollen mother cells, that is the PMC. That means PMC denotes the primary mother cells. The innermost layer of the cell wall that lies in contact with the primary mother cell, जो innermost layer है cell wall की, cell wall के अंदर further जो innermost layer होगी वो क्या करेगी, किसके कांटेक्ट में होगी, प्राइमरी मदर सेल के कांटेक्ट में होगी, जो कि क्या बनाएगी, टैपेटम, दैट फॉर्म्स द टैपेटम, एंड दिस टैपेटम इज सिग्निफिकेंट इन पॉलन डेवलपमेंट. और यही एक टैपेटम होता है जो किस चीज के लिए सिग्निफिकेंट होता है, फॉर द एंथर डेवलपमेंट के लिए. दैट मीन्स टैपेटम प्लेज एन इंपॉर्टेंट रोल इन द पॉलन डेवलपमेंट. जितनी भी पॉलन डेवलपमेंट होगी, दैट मीन्स दैट टेक्स प्लेस विद द हेल्प ऑफ द टैपेटम. What do you mean by tapetum? It is the tissue. Tapetum जो है वो एक तरह का टिश्यू है that is present within the anther. Tapetum जो है that is the type of the tissue and that is present in the anther and that provides the nourishment to the growing spores. क्योंकि tapetum जो है वो कहाँ पर प्रेजेंट है, एंथर में प्रेजेंट है, और वहां पर प्रेजेंट होने के बाद वो क्या करता है, वो प्रोवाइड करता है नरिशमेंट किसके लिए, टू द ग्रोइंग स्पोर्स. क्योंकि वहां पर जो भी स्पोर्स जो है, ये पोलन्स जो है जो ग्रो कर रहे हैं, दैट मींस दे नीड द नरिशमेंट विद द हेल्प ऑफ द tapetum, and after that nourishment that they need from the tapetum, after that they can be easily grow. The layer below the epidermis form the endothecium. जो लेयर हमारी एपिडर्मिस के नीचे है वो लेयर जो होती है वो क्या बनाती है, endothecium. This is the layer that lies below the epidermis and that layer is known as the endothecium. And this is the representation of the anther, structure of the anther. In the structure, this is the filament, and in the after the filament they are anthers, and in the anther there are number of pollen sacs that are present in the anther, and above the pollen sacs there are number of pollen grains that are present in this structure, that is the structure of the anther. Next is the wall layers of the anthers. How many wall layers of anthers are present in the flower? First is the epidermis, that is the wall layer of the anther, and epidermis that is a single layer of cells. epidermis में जो है सिंगल लेयर है सेल्स की एंड प्रोटेक्टिव इन फंक्शन, और ये जो लेयर है वो क्या करती है, प्रोटेक्ट करती है फ्रॉम आउटसाइड द मटेरियल. दैट इज दे हैव टू बी main role, that means the main important role that the epidermis plays, that is the protective in function. वो प्रोटेक्शन का काम करते हैं. नेक्स्ट इज द एंडोथीशियम, ये भी सिंगल लेयर सेकंड वॉल है. सेल्स हैव सेलुलोज, इसमें इसके जो सेल्स है दैट मींस दे हैव द सेलुलोज थिकनिंग विद द लिटिल पैक्टिन लिग्निन. इसमें जो सेलुलोज की जो लेयर है, इसमें जो मेन कंसेप्ट है जिसकी वजह से सेलुलोज की लेयर जो थिकनिंग है, that means they have little pectin lignin. It helps in the anther digestion. ये किस चीज में हेल्प करती है, anther digestion में. Next is the middle layer, ranges from 1 to 6. middle layer जो है वो 1 से लेकर 6 तक शुरू होती है. middle layer degenerates at the time of maturity, और ये middle layer जो होती है वो degenerate हो जाती है, टूट जाती है, ब्रेक आउट हो जाती है at the time of the maturity of the anther. जैसे ही anther mature होता है, at that time that middle layer is degenerate or break out. Next is the tapetum, innermost layer of the anther wall surrounding the sporogenous tissue. जो innermost layer wall है anther की, that is surrounding with the help of the sporogenous tissue. Tapetal cells are nutritive. जो tapetal cells होते हैं, जैसे कि अभी हमने कहा कि tapetum जो होता है that provides nourishment to the pollen sacs or the anther. That means it is the nutritive tapetal that plays an important role in the nutrition as well as nutritive or nutrition to the developing embryo. They are multinucleated and polyploid. ये multinucleated होते हैं, number of nucleus इसमें present होते हैं, polyploid होते हैं. the Ubish bodies are deposited in the exine of microspore wall. इसमें जो bodies होती हैं they are deposited in the exine of the microspore wall. Tapetum is of two types. Tapetum further हमारे पास two types का है. First is the secretory glandular, second is the amoeboid periplasmodial. तो secretory या glandular जो होता है tapetal cells वो remain in situ all through the development microspore and finally they degenerate. जो tapetal cells होते हैं वो क्या कहते हैं, मतलब development तक वो क्या रहते हैं वहां पर स्टोर रहते हैं. आफ्टर दैट दे फाइनली degenerate. अमीबॉइड और पेरिप्लाज्मोडियल जो होते हैं इसमें कैपिटल सेल जो होते हैं वो ब्रेक करते हैं थ्रू द रेडियल वॉल. इसमें भी कैपिटल सेल जो होंगे वो ब्रेक डाउन हो जाएंगे थ्रू द रेडियल वॉल, और जैसे वो ब्रेक डाउन होंगे तो वो क्या करेंगे, टू रिलीज द प्रोटोप्लास्ट इनटू द पॉलन चैम्बर. वहां से वो प्रोटोप्लास्ट को रिलीज करेंगे और प्रोटोप्लास्ट रिलीज करके वो किसमें उसको रिलीज करेंगे, पॉलन चैम्बर में. The protoplast now fuse to form the periplasmodium. अब जो प्रोटोप्लास्ट होगा वो फ्यूज कर जाएगा. फ्यूज करने के बाद वो क्या बनाएगा, periplasmodium. That is the final product that is formed after the fusion of the protoplast. Next is this is the diagrammatically representation of the tapetum or the anther. That is the structure of the anther. This is the filament, and after filament there are anthers, and in the anther there are number of anther filaments. They are attached with the help of the filament, and this is the transverse section of the anther. Transverse section of anther. This is the मिडल लेयर. जैसे कि हमने कहा था इसमें मिडल लेयर भी है, आउटरमोस्ट लेयर भी है एंड इनरमोस्ट लेयर भी है. एंड दिस इज द मिडल लेयर, एंड इन द मिडल लेयर देयर इज द tapetum. After the tapetum there is the sporogenous tissue, and after the sporogenous tissue this is the layer that is the endothecium. That means that lies under the middle layer. Middle layer के बाद जो layer होगी that is the endothecium. That means they have to be protected from the outside attacking material, and the outermost layer in the anther that is the epidermis, and this is the connective tissue that they have to be joined the tissues, and this is the B part of the structure of the anther. That is, this is the tapetum, these are the microspore mother cells, this is the middle layer, and after the middle layer this is the endothecium, and the outermost layer is the epidermis. So this is the structure of the anther or the transverse section of the anther. Next is microsporogenesis. What do you mean by microsporogenesis? The formation and differentiation of microspores. जो हम क्या करते हैं, फॉर्मेशन करते हैं माइक्रोस्पोर्स की, या फिर उन माइक्रोस्कोप्स की फॉर्मेशन होने के बाद जब उनकी डिफरेंशिएशन होती है उसको हम क्या कहेंगे, माइक्रोस्पोरोजेनेसिस. जो प्राइमरी मदर सेल्स होते हैं अंडरगोज म्योसिस. जितने भी प्राइमरी मदर सेल्स होंगे वो कौन सी प्रोसेस के अंदर जाएंगे, म्योसिस की प्रोसेस के अंदर जाएंगे, एंड ईच फॉर्म्स टेट्राहेड्रल टेट्राड्स. क्योंकि अब म्योसिस का प्रोसेस हो रहा है, फर्दर उसमें डिवीजन होगी, और जैसे ही डिवीजन होगी तो वो क्या बनाएंगे, टेट्राहेड्रल टेट्राड्स को बनाएंगे. साइटोकाइनेसिस में भी सक्सेसिव और साइमल्टेनियस. जो साइटोकाइनेसिस का प्रोसेस होगा दैट मे बी सक्सेसिव और साइमल्टेनियस. वो सक्सेसिव भी हो सकता है और साइमल्टेनियस भी हो सकता है. टेट्राड और ऑफ 5 टाइप्स. अब जो टेट्राड हमारे बने हैं दे आर फर्दर ऑफ 5 टाइप्स. टेट्राहेड्रल, आइसोबाइलेटरल, डीक्यूसेट, टी शेप लीनियर, टेट्राहेड्रल इज कॉमन. अब इसमें जो हमारे 5 हमने टाइप्स पढ़े, तो 5 टाइप्स में जो हमारी मोस्ट कॉमन टाइप होगी दैट इज द टेट्राहेड्रल. इन सक्सेसिव टाइप द सेल वॉल इज फॉर्म्ड आफ्टर म्योसिस. सक्सेसिव के केस में, सक्सेसिव टाइप के केस में जो सेल वॉल होगी वो कब बनेगी, आफ्टर द म्योसिस 1 एज वेल एज म्योसिस 2. तो मतलब हमारी जो सक्सेसिव टाइप में जो हमारी सेल वॉल बनेगी वो बनेगी आफ्टर म्योसिस 1 एंड आफ्टर म्योसिस 2, forming an isobilateral pollen tetrad, and it is characteristic feature of the monopod. That means main important characteristics of the monopods, and as all we know that monopods, that means they have single cotyledons in them, so these are known as the monopods. In simultaneous type, each nuclear division in microspore mother cell is formed by the cell wall formation. Simultaneous type में हमारी न्यूक्लियर डिवीजन जो होगी माइक्रोस्पोर मदर सेल से तो टेक प्लेस करेगी, पर वो टेक प्लेस करेगी दैट इज फॉलोड बाय द सेल वॉल फॉर्मेशन. जैसे ही सेल वॉल की फॉर्मेशन होगी, दैट मींस एट दैट टाइम न्यूक्लियर डिवीजन इन माइक्रोस्पोर मदर सेल्स takes place, and this is the diagrammatically representation of the microsporogenesis. This is the diagrammatically representation of the microsporogenesis. That means these are all pollen grains that occur in the microsporogenesis. This is the cells of the sporogenous tissues. These are meiosis, and in meiosis 4 cells, they are haploid. Anthers mature. Microsporogenesis, they stay together, वो इकट्ठे रहते हैं. Dehydration, dissociate, and the tetrad cells separates. और उसके बाद जो tetrad cells होंगे, that means they are separate with each other. This is the shapes of the tetrad, isobilateral, decussata, and linear, and T-shaped. So these are the 5 types of the cells that are present in the microsporogenesis. The microspores are separated from the tetrahedral arrangement. जो microspores हैं वो separate होंगे, अलग हो जाएंगे, पर वो अलग होंगे with the help of the tetrahedral arrangement. They are then surrounded by a layered wall. उसके बाद उसको surround किया जाएगा 2 layered wall के साथ. Outer wall जो भी उसको हम exine बोलेंगे, और जो inner layer होगी उसको हम क्या बोलेंगे, intine. अंदर वाली layer होगी that is known as intine, and जो outermost layer होगी that is known as exine. Pollen grains are the first cells of the male gametophyte. अब जो हमारी pollen grains develop हो चुकी है, grow हो चुकी हैं, that means pollen grains are the first cells. अब वो first cell बने किस चीज के, male gametophyte के. Tapetum gets used up. अब tapetum को use up किया जाएगा and the anther becomes dry structure. Tapetum को जैसे ही use करेंगे, use करने के बाद जो anther होंगे वो कैसे हो जाएंगे, उनका structure dry हो जाएगा. This liberates the pollen by digestion of the anther. उसके बाद वो pollen को क्या करेंगे, liberate करेंगे by the process of digestion of the anther. All the 4 nuclei in the tetrad remain functional. जितने भी हमारे nuclei हैं tetrad में, वो क्या होंगे, functional होंगे, और functional होने के बाद वो क्या बनाएंगे further 4 microspores. In some cases, all the 4 pollen grains attach. कुछ cases में 4 pollen grains होंगे वो attach करेंगे, forming compound pollen grains. For example, Juncus jatropha. This is the common example of the pollen grains. In family, that is Asclepiadaceae and Orchidaceae, microspores are present as Pollinium. इसमें जो हमारी फैमिली बताई हमने Asclepiadaceae और Orchidaceae, इसमें माइक्रोस्पोर्स होंगे, they are present as a Pollinium. अब pollinium क्या होता है? It is a mass of pollen grains that is present at the anther lobe. ये एक तरह का mass है pollen grains का, जो कहाँ present होगा, anther lobe पर present होगा. As the pollinium is attached, जैसे ही pollinium attach करेगा pollinating agents के साथ, अब pollinating agents जो हैं, that means insects, entire mass of the pollen grain that is transferred as a unit. Next, pollen grains. pollen grains vary shape. जितनी भी pollen grains होंगी, हर किसी की shape जो होगी वो अलग होगी, vary करेगी एक दूसरे के साथ. It is generally rounded with size of 25 to 30 म्यूसीमीटर. इनका जो generally round size जो होता है वो 25 से 30 म्यूसीमीटर के बीच में है. pollen grains जो होते हैं वो haploid होते हैं. That means they have single unicellular single cell body, इनकी single nucleus होता है, 2 layered walls इनमें. Wall of the sporoderm consists of 2 layers. जो wall है sporoderm की, that consists of the 2 layers. Outer layer जो है thick है, that is called exine, and it is made up of sporopollenin. Inner wall is thin and that is called intine, and it is made up of pectocellulose. जो intine की layer है, that is made up of pectocellulose. The exine is thick and sculpted or smooth. जो आपके exine होती है वो thick है और smooth होती है. Cutinized, and cutin is of special type called sporophyll lineage, which is resistant to chemical and biological decomposition. This prevents the pollen wall for long periods. और ये जो pollen wall को है वो काफी लंबे समय तक preserve करती है. It also possesses proteins भी होते हैं इसमें for enzymatic as well as compatibility reactions. Exine is differentiated into inner endexine and outer extine. तो ectexine is further divided into inner continuous foot layer, middle discontinuous baculate layer and outermost discontinuous tectum. Tectum aid is the identification of pollen grains and assigning them to their family, genus or species. Pollen grains contains regions of pores or furrows, and in these regions exine is absent. So on the behalf of that reason we can say that exine is absent in this region. Intine is thin and elastic. Intine जो है वो thin होता है और elastic है. It is made up of cellulose and pectin. अब thin और elastic है, उसके बावजूद उसकी जो layer है वो किस चीज की बनी हुई है? Cellulose and pectin की. During pollen germination, जैसे ही pollen germination होती है, it is the intine that extends out to form the pollen tube. The cytoplasm of the pollen grains is rich in starch. जो pollen tube है, that is rich in the starch and unsaturated oils. They are initially uninucleate and later becomes 2-3 cell. In calotropis and orchids, the pollen of each anther lobe formed a characteristic mass called pollen. Pollen grains can be monocolopate having 1 germ pore. Monocolopate means that they have 1 germ pore, bicolopate 2 germ pores, tricolopate 3 germ pores. The branch of study of pollens is called as palynology. जो branch है हमारी study किस चीज की? Pollens की, that is known as the palynology. So this is all about the pollen germination that takes place in the flower, and the next topics to be covered in the next lecture.